Okay, so now we're moving on to part 2 of chapter 14, or part B as I have it listed on your note-taking sheet. So the first question is, how do tumor suppressor genes work? What do they do? What does it mean? Well, the wild-type genes that are tumor suppressors, they prevent unregulated cell growth, preventing cancer. Now, as you'll see later, there's a couple of different mechanisms by which different tumor suppressor genes can work to prevent cancer. Now, we left off last time talking about oncogenes, and I wanted to give you a couple of specific oncogenes because these are very well studied and known and understood. And they also become oncogenes from proto-oncogenes by different mechanisms. The RAS oncogene is involved in regulating cell division. The RAS protein transmits a signal from the cell surface to the machinery inside the cell that regulates cell growth. Normally, a signal arrives. When that happens, the RAS protein binds to GTP, and that is then translated into a signal that goes off to the growth machinery. Now, when it breaks down GTP, hydrolyzes it to GDP, the signal is turned off. And that happens from a region that's actually part of the RAS. So RAS will bind the GTP and then send a signal, and then it turns itself off. Now, the proto-oncogene, which is great, very useful, becomes an oncogene when we get a mutation in that region that's involved in turning off the signal. So GTP doesn't get converted to GDP, it stays stuck with GTP, and that means RAS can't turn off, so it just starts telling the cell, grow, grow, grow. You should really understand this figure. It basically goes over what I was just describing, but you should be able to come up with a conceptual framework that would match this figure. Now, the MYC oncogene gets turned into the MYC oncogene gets activated in a different mechanism. So the proto-oncogene for MYC is a transcription factor. It means it's involved in regulating the mRNA production of other genes. This is a good thing. I mean, without it, we'd probably be dead. The problem is, every now and then we'll get overproduction of the MYC protein. Now, that can be due to one of two different mechanisms. One could be gene duplication, and the other is chromosomal rearrangements, which increase the expression of the gene, so that you end up with a whole being put in front of a strong promoter, and you get a whole bunch of mRNA, and you end up making more MYC protein than you should. So it's just a real quick picture. It just shows the mechanism for gene duplication, so you end up with copy after copy being made. Even 1 or 2 extra copies could be bad. Here they show 4. Another possibility is that it can be moved from one chromosome to another chromosome. These kinds of things do happen. Things do get swapped around through homologous recombination and so forth, and then sometimes mistakes are made. And so for this mechanism, what ends up happening is the proto-oncogene gets put in front of a strong promoter, which then drives a bunch of mRNA synthesis, which causes a whole bunch of protein to be made, and then you have problems because the MYC protein is also one of those proteins that's involved in saying grow. Normally, there's just a normal amount, and it doesn't cause a problem. But when you've got tons of it, the signal is grow, 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 and it doesn't stop. Now, proto-oncogenes kind of break down into 4 different functional groups. The first are growth factors. Now, growth factors are circulating proteins. They bring information to the cell surface receptors. Cell surface receptors are proteins that are found in the plasma membrane, and they'll take that information and transmit it from the outside to the inside of the cell. Guess what? The RAS protein is one of these. Transcription factors, which are found in the nucleus that regulate mRNA production. We just talked about one of those. The MYC protein is a transcription factor. And finally, there are protein kinases. Now, protein kinases we'll talk about on the next slide because they're a little more complicated. So you go to a signal that comes from the outside of the cell. It often activates a protein kinase inside the cell. So what does a protein kinase do? A protein kinase is an enzyme that transfers a phosphate from ATP to a protein. Now, when that happens, when the protein now has a phosphate attached to it, it may turn off the function of that protein. It may turn on the function of that protein. So the kinase is regulating the protein's function. Protein kinases like I said, change the function of other proteins inside the cell in response to a signal that's transmitted from the outside typically. Okay, so tumor suppressor genes. The function of both copies of a tumor suppressor gene must be lost due to mutation in order to cause cancer. All right? If you've got one good copy, usually, you're going to see there's an exception, but usually if you've got one good copy, you're fine. So typically both copies have to actually be inactivated in order to cause cancer. So a tumor suppressor gene is basically preventing this unregulated cell growth, but if you lose a function of a tumor suppressor gene, that check is lost, and therefore you may end up developing cancer. So the loss of function of tumor suppressor genes, there's kind of two ways. I mean, they're not really distinct ways, they're just different probabilities. Very rarely you'll get two somatic mutations in the same gene in the same cell. Now what this means is you were born normal, both your copies of the tumor suppressor gene that we're talking about here are normal. Somehow during cell division, both copies developed mutations that inactivate the gene. Now this is really rare, okay? More commonly, you inherit one bad gene from a parent. And then the probability of the second gene acquiring a mutation, is much higher. So this has been studied in retinoblastoma, which is basically cancer of the retina in your eye. In healthy individuals, fine. If they get one mutation, nothing happens because they've still got a good copy. In hereditary retinoblastoma, you start out with an individual who's got one mutation, and then if you end up with a much higher probability that you might pick up one mutation in the other chromosomal copy. If that happens, most people are going to develop a tumor. However, this non-hereditary form, where you got to get two mutations that happen, is really rare. It's been studied. It's about 1 in 30,000 people will develop a tumor. So it's very unlikely if you have a normal retinoblastoma gene that you'll end up with this particular issue. Now, how do tumor suppressors work? Well, there's kind of two theories, all right? And I think they probably are a little beyond theory at this point. I wouldn't call them laws yet, but basically the idea is tumor suppressors work by doing a couple things. One mechanism by which they work, depending on the tumor suppressor protein, is they're involved in slowing down the cell cycle. In other words, stopping things so that you don't end up going crazy and growing. So they'll basically sort of damper down signals that come to the cell surface that get translated inside the cell that are then involved in telling the cell to grow. So it kind of slows cell growth. The other thing is some tumor suppressor genes have been found to be involved in pre-programmed cell death. Now, what do I mean by that? You may or may not have heard this term apoptosis. That's another parallel term and synonym that means pre-programmed cell death. So cells have a predetermined number of divisions. This is known, all right? So one particular cell in your body might only grow and divide 40 times and it will die. How does it die? Well, these tumor suppressor genes that are involved in pre-programmed cell death, basically somehow they sense that the cell has divided so many times, and they turn on this apoptosis pathway, which basically causes the cell to kill itself. So when a tumor suppressor gene is nullozygous, that's a fancy word, it just means homozygous recessive, the death signal's gone. So if that signal's gone, cells that normally maybe would die after 40 cell divisions don't die, and they keep growing. One piece of logic behind this is if you can cause the cells to kill themselves after 40 cell divisions, all the accumulated mutations will be gone, essentially. And so new cells will come from germline cells, which hopefully are not going to be, mutant or whatever. And so you'll start with sort of a fresh set of 40 cell cycles to be, to go back through and have the cells, do the normal function for 40 more cell cycles. Now, what is a cell cycle? You guys had this in biology, all right? So it's nothing new. The cell cycle has been really well studied. Basically, it's how the growth of a cell is controlled, okay? So you've all heard about things like mitosis and G1 and G2, but I'm going to go back through this a little bit. There's four main parts. G1 is growth phase number one. Now, the cell sort of senses, oh, yes, I've got enough food, and so it commits to starting to get bigger and bigger. There's a checkpoint called the G1S transition. It's controlled by cyclin E, and we'll talk more about cyclins in a minute. If it gets big enough and the environment sensed is such that we've got plenty of nutrients out there, the cell will then commit to S phase. Now, S phase is where DNA is made, so you're going to duplicate your chromosomes during S phase. Now, we have another important checkpoint called the SG2 checkpoint. So going from S phase to G2, which is the second growth phase. Cyclin A regulates this particular step. All right, so once you commit to G2, you commit to growing bigger and bigger and bigger. All right, so that's your growth phase number two. Now, once you're big enough, there's one final checkpoint. That's the G2M checkpoint, and that's controlled by cyclin B. So G2M basically is going from the growth phase to committing to mitosis. And then finally, the cell undergoes mitosis, the nucleus and the cell divide. Now, cyclins are proteins that somehow can sense things that will allow the cell cycle to progress. If they sense, like for instance, cyclin E senses, oh yeah, there's plenty of nutrients around, yeah, we can commit to duplicating our DNA, cyclin E will bind to a cyclin-dependent kinase, activating it, and that kinase will phosphorylate a bunch of proteins, and the net result is it will turn things on, so it will commit to duplicating the DNA. Cyclin A and cyclin B work by the same mechanism. Cyclin proteins can sense things. There isn't enough detail here to explain what they sense, but then once they seems like everything is good, they will activate a kinase, and that causes the next round of, or next stage in progression of cell growth. And I think that is it. Oh, nope, one last slide. Okay, so this just kind of shows a rough idea of the amount of time it takes for a cell. Now, this is very arbitrary, okay? It says 12 hours. Human cells typically don't grow and divide in 12 hours. Some cells may in you, but a lot of the cells grow and divide over days or weeks. So it depends on the cell type. If it was a bacterial cell, this whole cell cycle takes about 20 minutes. If it's a yeast cell, the whole cell cycle takes about 2 to 3 hours. So the steps, though, in the progression are pretty much the same in all eukaryotes. It's just that there's timing differences. And this just shows also the sort of the organization of where the cyclins are, so you can see where the control points are.